ท่านรองศาสตราจารย์นายแพทย์ดิเรกครับ
Speak English because we need to use some technical term. We maybe give you give the translator a hard time. So I'm sorry about that. And first of all, I would like to thank for inviting, and I will talk about the AMR situation in Southeast Asia. I will also touch a little bit about the antimicrobial use in the region, and also some work that has been done in the region as well. It doesn't move. It's my bad. I pushed the wrong button. Okay, so I think this is a room that we don't have to introduce about AMR anymore. But for everyone here, AMR is a shared concern, it's a shared issue, and AMR doesn't recognize the geographic regions, and it doesn't have the border between human, animals, and environment. So to address the rising of AMR threat, it requires a holistic and multi-sectoral action, which is AMR which is a One Health approach. They are the AMR control keys that have been suggested to be applied for other countries at national, international, or even regional level. But for this, you can see there are many keys that we can include, like, for the first one, antimicrobial use and antimicrobial resistance surveillance, up until the number 6, which is research intervention. It looks very nice on the pictures. It looks very nice on these slides, but for real, it's not that easy to implement. So this is just a little overview about Southeast Asia. Okay, Southeast Asia has about approximately 700 million of the population. The country with the largest population is Indonesia, and that with the lowest one is Brunei. If Asians were a single unit, it would rank as the third most populous in the whole world, and is ranked as the third largest economy in Asia, and it is the 7th largest economy in the world. Most countries in Southeast Asia are relying on agriculture and farming. Okay, and the most important or the most common livestock productions are poultry, including chickens and ducks, and also followed by the buffalo. And the low ones, there were also some, little bit of the goat. But in some countries like Singapore and Brunei, the food animal production is not a major part, and food of animal origins are mostly reliant on the importation. And for our laboratory, we work as a team, we work as a research unit. So we have done many kinds or many projects in the area of AMR, and we use the AMR and One Health approach as our base of the research. This is what the topic in general. We do some kinds of research about phenotypic and genotypic epidemiology of AMR. We also do study about mechanism of AMR. Some of our team do the risk assessment of AMR. We also try to figure out the link between antimicrobial use and antimicrobial resistance. And also some of us also work with the alternatives to antibiotics. And today what I will introduce you is the first one, phenotypic and genotypic epidemiology of AMR. At the beginning, when we started to work, and graduated, I will not tell you how many years, otherwise you will know how old I am. But at the beginning, I started in the country first, and now we know that AMR is borderless. So I expand my work to the other country in the same region. So now we can work with AMR in Myanmar, in Laos, and also in Cambodia. We do hope that we can expand our research, to the whole Southeast Asia. And AMR is a growing issue in Southeast Asia. This data is from our previous review that was done for FAO. This data I will accept that it's a little bit outdated and it should be updated. So we have to do the review for the review. Okay. So for this, what you can see, most of the studies, in Southeast Asia focus on Salmonella and E. coli, and this is not surprising because Salmonella is one of the major foodborne pathogens in this region, and E. coli is the commensal bacterium, and is an indicator of contamination in food. Okay, and one more thing, some bacteria, these are fastidious bacteria like Campylobacter. The technique to isolate these bacteria are not that easy, so that could limit the research in these regions as well. But based on the data that we gather, we can see that the prevalence of resistance to the basic antibiotics are quite high, but it's low in certain antibiotics. And this is not surprising as well because this antibiotics has been extensively used in food animal production in the region. But when we take a look to the particular species, so take a look in Salmonella first. Okay, what we can see, we can see the different resistance rate for different antibiotics and in different countries. Okay, so like for example, look at the tetracycline here, right? The resistance rate for tetracycline is pretty high in most countries. It is the same for nalidixic acid, okay? But this is not the case for like ciprofloxacin, which is quite low. So one more thing that we have to keep in mind, this information may be influenced by the difference of the technique, the difference of the sampling technique, and also the difference of breakpoint that have been used. And this data has been published, or it was done before the time that we encourage the use of harmonized and standardized methods for antimicrobial susceptibility testing. And here for E. coli, it's not different, it's similar, okay? So we have to accept that we really don't know what the source of this E. coli. For AMR monitoring, we would like to include just only the E. coli from intestinal tract, which is commensal E. coli. So, but this one we cannot guarantee that all the E. coli were from intestinal tract or they were commensals. But if we think that these E. coli were from intestinal tract, they were commensal from healthy animals, these resistance rates will represent the antimicrobial use in the food animal production in the region. And everyone here, interested in ESBL, I think is another concern and is another topic of the research, okay? So we also work with ESBL. This is the data from another project. This project is on ESBL production of the bacteria associated with food animals in Thailand and neighboring country. And the neighboring country that we include were Laos, Cambodia, and Myanmar. Please be noted that this is from the border provinces, not the whole country. But what we can see here, Myanmar, which is the lowest one, has the highest prevalence of ESBL, which is 11. But anyway, when you look at the whole picture, the prevalence of ESBL producing E. coli is still low. And here for Salmonella, we tested for ESBL producing Salmonella as well, and we can see the same scenario that the ESBL producing Salmonella is still at very low rate, and for Laos you can see that it's even 0. Okay, for those is the data from the province at the border area, but this is come to the center of Thailand, okay? So we did some experiments about ESBL productions and colistin resistance in commensal E. coli from pigs. We did the experiment for 2 types of pigs. The first one, look at the cartoon, you can see that he's sick. So this is the isolate from the sick pig. And this one you can see the smile with the pink face, so he is healthy. So we did the experiment in the pig that were clinically sick and clinically healthy. Well, so what we did test for the ESBL and colistin, and we found that The prevalence of ESBL producing E. coli and those resistant to colistin is higher in the isolate form clinically sick pigs. And this is not surprising as well because these sick pigs were expected to get some antibiotic treatment. And also we did some tests for the genes, MCR1 and MCR2. I think everyone in Thailand know this because it was famous in the news. It was even famous than the superstar in the early of last year, right? We saw the story about MCR1 in the newspaper, and we found that all of the colistin-resistant isolate from sick pig contain MCR1. But this is not the case for the isolate from healthy pig, okay? So this one, okay, you can see only 7.3% of the isolate from healthy pig carry MCR1. This implies us that there are some other mechanisms existing and involved in the colistin resistance in this isolate. And we also tested for the MCR2, and we found only one isolate was positive to MCR2. And we're now working on MCR3 and MCR4. Fortunately, we don't see any of them. And we also observed that those bacteria that produce ESBL are also resistant to colistin. So what we did some experiment to transfer the gene. So we did tests called biparental mating. Okay, biparental mating that transfer the plasmid that carry both genes to from E. coli to Salmonella. And we found that we could transfer the plasmid carrying both ESBL genes and MCR1 on the same plasmid. And the ESBL gene that we found is what BLA-CTXM, and it could transfer the resistant phenotype as well. So this is the possible schematic structure of the plasmid carrying BLA-CTXM and MCR1 gene, and we are in the progress to do the plasmid mapping. And AMU and AMR are close friends. They support each other, so we would like to destroy their relationship. And for AMU in livestock in Southeast Asia, we cannot say that we don't know everything. We know something, but it's still unclear. Types and extent or the amount of antimicrobial that used in this region is different from one country to another, depend on the development level, veterinary practice, and also their economy. But in general, the regulation of antimicrobial use in this region is still not that great. Even though in some countries there is the existing regulation, but it is poorly enforced. And most of the country doesn't have national antimicrobial use surveillance. Actually, some countries already started the surveillance, though I think from what I know, including Thailand, the Philippines, Singapore, that already started the surveillance for the antimicrobial use. And the thing even worse, because we have the backyard farm or smallholders that are still common, and some of the farmers can still access, the antibiotic over the counters. And this is a challenge for the government that have to do something to stop this behavior. And fortunately, right now, there is the guideline. This is a guideline, it's an ASEAN guideline for antimicrobial use that has been launched. It was drafted, or it was written by Singapore, which is the leading country of AMR in Southeast Asia. This is already endorsed by ASWGL. Don't ask me what it is, it's so long, okay? And this one was intended to be the guide for any country that would like to draft the guideline for antimicrobial use. I know now that Singapore is using this one as a guideline to draft their national policy. And from my understanding, Philippines we also use this guideline, but I don't have the update, for the other country. And here to the last one, which is the work that we have done about AMR. Okay, the first column is about our history. Okay, right now, well, we have changed many names. And right now we are CU with AMR, not AMR, which is Chulalongkorn University Veterinary Antimicrobial Resistance Cluster, and we have a research unit at the Center for Antimicrobial Resistance. Okay, our work, has been started with for AMR, had been in collaboration with many organizations, but the most of the organization that we collaborate most is the FAO. So we started with the work with FAO, in 2013. We did the first work. It was about the review. We did the review for AMR in Asia Pacific first, and then go to the global level. And we did some antimicrobial susceptibility training. And in 2016, we started the assessment of AMR laboratory capacity for Southeast Asia, and this one has gone very well. We did it also in 2017. I think we finished about 7 countries, and the result, I think, is up on the request. You can request it from FAO. And we also did the training, and we also adopted the guideline for AMR surveillance in bacteria associated with food animals. So right now in FAO hands, so they wait for them to endorse. And here, at this moment, we are awaiting formal recognition as FAO Reference Center for Antimicrobial Resistance Monitoring. The role will be for Asia Pacific. And the work plan has been set, so hopefully that in the future we can do something more for the region and would also support the global plan for the AMR. That's pretty much it. Thank you.